Hello again, that's me. I hope you have watched the first introduction to the basics of XMAS. So I will start over where I left, again with the same structure and the same set of evidence files. This is fibrinogen. What do we know about these proteins? A bit that forms clots in the blood, but maybe we are missing something. So if you click on the links, it's not up, but can look into uniprods for this protein. Of course, I did already, but let's do it again. So it's fibrinogen, the alpha chain. So this is the uniprot ID. If you copy paste this one and you fetch, so the command is really AlphaFold fetch E0. It will just fetch just changing this right mouse. So when we see, so this is the full sequence. Of course, it has some transit peptide, fibrinopeptide, but it's clear that most of it's not represented in the structure. So if we do matchmaker, so this is also very basic commands. When you match one model to another, you will see that it is matching for the right chain, but we have still many missing parts. So how could we do to complete the structures? Do we have evidences based on cross-linking MS data that links these additional models to our basic structures? So we just need to include these two models and map the evidence files. We start with the single replicates and we map the crosslinks. Wow, this is really massive. But why? Because it will start now to try to connect every possible combination of this complete alpha chains to this other model. Good thing is that we can now then filter this out. But actually, we're not interested in the full structure, but only in some of the domains. So what we can do, for example, is let's say we have this loop going from 220 to, for example, this residue here, 264. So what we can do is to select, so we go to tools, sequence, show sequence viewer, and we want to see the sequence of our AlphaFold model for the alpha chain. Here we go. We have these two amino acids, histidine and proline, that sends the beginning and the ends of this small loops that is not present in the structure, but might have some data for it. So what we want to do is to actually select whatever it is, not this part of the sequence, and you can either delete the selection by commands on the command line, or you can also do the edit version. Now we deleted the first part. Want to delete the N termini. This. Here we go. We only have left this small chunk. So let's do this again. So we map the crosslinks. And boom. And now we start to see crosslinks of this little guy here, which is part of the complex and the complex itself. We can do this for all the replicates in a few clicks. And then I will show you later how we can use this data. 1, 2, and 3. You can close the sequence here. Okay, here we go. So first thing we can start to do is actually exporting. So we select our 3 replicates, we export, and in this case we want to export only the interlinks between the models. So we want to export the crosslinks that are helping us to dock this additional domain of the alpha chain to the core model. And we want to do this with highly reproducible crosslinks, let's say 3 out of 3 pseudobond models. And we want to export for a tool which is called DisVis, which will do the occupancy analysis based on the distance restraints. So in one go, we can just export. We don't care about the distance now, because where we place this small stretch of the protein is arbitrary. But we want to do this based on the crosslink and use them as distance constraints. So the fixed chain is let's say usually the bigger protein, and the scanning chain is usually the smaller, but you can do this with dimers, for example. The minimum and maximum distance we want to set, they are defined by the crosslinker. We can allow a little bit of flexibility here. Let's say we stay in the canonical ones. This is for Fox, but with DSS or DSSO, you can just, or zero line crosslink, you can define any arbitrary but reasonable minimum and maximum distance. When you press OK, what it will do in this tutorial, we go to integrative modeling, Fibrinogen, and then we give the name, and it will output a different file. So we call it module doc, save it. So if you have the pseudobond models, which is defined as only the interlink between our selected additional small domain of the protein and the main fibrin core. Now, what we want to do is to go on DisVis. DisVis is a web server. It's in here. Let's go, and here we can submit. So what we have, okay, you can also have a local version of this. We call it fibrin, and then we have already all the files we need. So if we go to folder where we saved everything, we have the chains. So in this case, we have our fibrin model as the fixed chain. Our scanning chain will actually be these small parts that we selected. And we have a restraint file, which is this text document. And I can show you how it is formatted, but for now we just do it like this, and we launch this. It depends on the size of the protein. It can take more or less time. Hello, so I switched on and off in order for the run to finish. In this case, it's very fast. In the DisVis web page, you will have a result. To upload to XMAS, you will need the complete run. So you can click on this link to download the folder. In the meantime, we can look at the results. So you will see here, so what this tool is doing, it's taking your scanning chain and then doing a rotational sampling around your fixed molecule using the restraints, and then it will give you an idea of the number of accessible complexes that are accessible with this number of restraints. So in this case, it was very fast. We only had 10 restraints. And then here you will see the Z score, that this is what we will actually visualize in XMAS. So let's go to XMAS. I can show you, you can start from a completely new session. In the integrate button here, you can analyze DisVis output, and then you go there and select the folder with the results. In this case, I downloaded it already. So you just select the folder, and then you hit this use folder button. So this will automatically open the structures to the restraints, and then you can visualize them as crosslinks, and volume. So what this volume is, you can play around with these levels you see here in the volume viewer. These are the number of the position of the center of mass of your protein. If you select it with the right button of your mouse, you can actually move it towards this center of mass. We can also make the volume transparent in a way that you can see what happens. So what you can see is that when we move it, by the time these restraints they become within the maximum allowed range that we set, so in this case was 5 to 25 angstroms, they turn gold or yellow. Otherwise they stay red, and this is quite interesting. It's quite useful to try to understand where your protein is placed, but we can do more. So here you see the Z score, which is it's to take with a grain of salt, but in principle, so this score is inverted. So let's say below 0.5, you can also slide it to see what's the score from DisVis, but you can see that this score it's giving you an idea of the good restraints you want to keep. So let's say we stay at 0.37. So in this case, you will see that some of them are still violated, but this is because we're just looking at the center of the mass. It's not a proper docking yet. So when we are happy, we hit the OK button, and you will see a new model with the restraints selected will appear in your models, and this will only contains the one that we filtered out. So in this case, it's we call them true positives. But so just to refrain, you can have different binding positions. So sometimes DisVis can point you to two different sites. We'll show you another example later. But let's say that now we are happy with this and we want to do the proper molecular docking. So we go back to the integrate button, and then we can create an HADDOCK input. We want to select the pseudobond model, but the one we filtered, And now it will ask you to select chain A. So in this case, we want A to be the fixed chain. Input types, we want the number of the residues, a restraints file, and the PDB file. We can use preset distance restraints. In this case, we need to put 3. So we can use the preset ones, in this case 5 to 25. Median is the expected length or distances, but in principle you should use again the ones defined by your crosslinker, but you can also play around a bit with this. So when you click OK, it will ask you to select the folder where you want to have this. Let's say we can call it HADDOCK inputs, and we can make a new folder. Here it will tell you the residues that are involved in this interaction. So now we can actually go to HADDOCK. So HADDOCK is another suite. It's quite popular from the Bonvin lab. HADDOCK 2.4. Here you need to register to get token, but it's free. And then we can submit, and now we made the job really easy. So we can call it test again. We have 2 molecules, and in the folder you will find everything that is necessary to run this. So we go to HADDOCK input, you will see you have chain A, chain B, and the selected restraints. So chain A, we have it. For a first test, you can use the preset settings. If you are not an expert user, I strongly recommend you to stick to the presets. This we can skip. So we do, if you want, you can define active and passive residues. So in this case, we can either choose to define these residues, so which is a list of residues involved in the interaction, and you can retrieve this information from here. So you can copy paste this line. So this 99, 112, this is the residues involved from chain A, and we can paste it here. Or we can, if you click next, this allows you here to supply, here on the second line, HADDOCK restraint TBL file with restraints that will be enforced, unambiguous restraints, or active and passive residues, again on this TBL file. What we can do is we can choose this restraints file. This is a TBL file that is automatically generated in your folder. And the format, it's a little bit different. I can actually show you how it looks like. But it defines basically the distances that we defined before. So 10 is the median, 5, and then it names the residues and assign these restraints ambiguously. So I think we selected this. Yeah, this is selected. And that's it. You can submit. Hello, everyone. I'm here for another example on how to use XMAS for your integrative modeling pipeline. So this time I will open a slightly different type of structure. So we have 2 small molecules. For this you can check we actually have a preprint out. So what we have here is factor 11, which is a coagulation factor in your blood, and thrombin, which is an enzyme that cleaves. And it's necessary to start the polymerization of your blood clots. So we can map crosslinks on this. For this, we have, for example, 4 different replicates that we can open here, and we just hit the button, map crosslinks, and we're interested in the interlinks between thrombin and factor 11, so in their interaction sites. So we redo all the pipeline again. We only look at interlinks for now. You will see that many conformational changes can come. One important part, it's always check, even if you map 2 monomers, check your table, so the TSV file. You can go back there, factor 11. And then we drag it in. Then I will show you why is that. So in this overlap category, we can filter. Okay, you will have, in this case, we have overlapping, non-self, and self. What overlapping self means? So if you look at the residue number, it's 547 and 547, which means that we have exactly the same residue cross-linked in both. And then we also have an overlapping peptide, which means that also these two point out to a homodimeric interface. And this we can actually model in XMAS. So we reopen now to make it faster. I will show you how to open again the DisVis output. So I run this already. We can select again the same, so factor 11, and all these files you can find them in the supplementary information. I will provide a link to the preprint, and you will find all the structure and the restraints if you want to try and play around. Use folder, and here we go. So what we have here, it's I think it's pretty obvious. So we have factor 11 dimer. We have interaction accessible interaction space that is defined by DisVis, and our set of restraints. So of course these are been selected. We can move the model. It's pretty obvious now where the interaction site is, but if you look at the map, you will notice that, so if we reduce, it points out to a circle around it, because in the end we have a very defined point, but our molecule could be accessible from different sites, and all of these are actually compatible with the distance restraints we put. So then we need to go through a round of molecular docking. So this will take into account the property of the amino acids and try to form an interaction in this interface that is stable between the two. So this is a pre-filter. So we can use again this z-score to remove, let's say, the false positives, or let's say the true positives that we want to keep and use for the docking. And again, we can hit OK. Then you will have the name of the pseudo bond file with the selected ones. And then again, we can create HADDOCK inputs with the selected ones. We select chain A. In this case, we want to select factor 11. Dimer, we define the distance restraints. We use the same linker, so we can actually use the same. We select the folder where we want this to be, and then you can see again we have the residue numbers of chain A and B. So with this, we go back to our HADDOCK and factor 11 tests chain A. Well, here we are still in the fibrinogen. Chain A, yes. And then we choose our chain B. That will be actually named chain B. So this is quite straightforward. Sorry. We can define the active residues or input these ambiguous restraints file. So in this case, we can choose TBL. It will be the file order, submit, and the run is true. So this run takes a bit more time, but again, you will have clusters out. You can follow tutorials and test HADDOCK, and happy docking to all of you.